So it's really my pleasure to be able to speak with Dr. Tim Reddy. Thanks so much for making the time. Dr. Reddy is Associate Professor of Biostatistics and Bioinformatics and Associate Professor in Molecular Genetics and Microbiology, and is a faculty member in the Center for Genomic and Computational Biology, as well as the Center for Advanced Genomic Technologies. Really great to have you. Thanks so much, Tim.
Happy to be here.
So Tim, you've been doing incredible work in understanding the so-called dark genome, the non-coding genome. And you've really been a leader in the ENCODE project for over a decade, I think. Can you tell us a little bit about the ENCODE project and sort of give us an update of what's going on with that and your work in it?
Sure, absolutely. Yeah, so the ENCODE project's been around, it's wrapping up now, it's in its final days, but it's a project that's been around 20 years now. It really came right on the heels of the sort of initial draft sequencing of the human genome, where we suddenly had this, this enormous 3.3 billion letter genome sequence, and the vast majority of it, we had absolutely no idea what was going on. And so the goal of ENCODE, sort of from its inception, was to try to fill in all of those gaps, to try to fill in what, understand what are all the parts of the genome that are contributing to human biology. The project has gone in so many different directions over the last 20 years. It's really been incredible. It started as an initial focus look at just 1% of the genome. When high throughput sequencing started happening, it became a genome-wide effort very rapidly. And for the past 15 years, everything has been really, a lot of it's been really genome-wide. ENCODE has always played an interesting, or sort of had an interesting position within the research community. Sometimes it has the investigators that are contributing to ENCODE have helped to develop some of the technologies like ChIP-seq and RNA-seq that we now rely on in our day-to-day research to understand how the genome's working. And at the same time, the consortium has also done a really good job of bringing in technologies that others have developed and either using them directly or in some cases sort of industrializing them to some extent so they can be deployed across a large diversity of different primary cells, tissues, and cell models, for example. And that's really where the growth of ENCODE has been, certainly in the last 10 years, and especially in the last 5 years, has been sort of an enormous push into primary tissues, primary cells. There's many consortium that have done similar work in that regard. There's, for example, GTEx has looked at gene expression across lots of different human tissues. But what ENCODE's been really especially good at are what we think of as these like the sort of multi-omic measures, where you're not just looking at gene expression in a cell or a tissue, but also the chromatin state and the transcription factors that are binding and the chromatin loops and how genes are being turned on and off and the dynamics of gene expression. And so what you end up with is a much more sort of holistic, complete understanding of how these different cells and tissues might be acting, or how the genome is contributing to those cells. With the goal of that kind of providing, if you will, a blueprint for how we understand, human biology. Not just human biology, I'll say also mouse biology. There's been several model organism components as well. What as we're wrapping up this final phase of ENCODE now, a number of the things that we're highlighting, one is that like I said, first of all, the diversity of tissues and cells and cell systems and organoids that we're able to now provide information on is just absolutely beyond where we've ever been. I mean, I think we've doubled or tripled since the previous phases of ENCODE, the amount and diversity of those cells and tissues. We've got much more increased resolution on how the genome is interacting. So these interaction maps of how one part of the genome connects to another. I mean, I've been seeing data sets where the resolution is down to 10 or 15 base pairs, and I can see enhancers touching promoters for the first time. I've seen the looping in the globin locus that I've never been able to see before at, where you can pick out every single little binding site. It's really incredible the amount of resolution that we're getting at that scale. I think that's really impactful for us. There's been a large effort in single cell technologies with the newest version of ENCODE. So there's hundreds and hundreds of these multi-omic samples where you basically take a cell, you use part of the cell to measure gene expression, the other part of the cell to look at the state of the genome. And then with that paired together, you can start to understand what genomic compartments genomic parts of the genome are contributing to the regulation of which genes in these like rare cell types that are buried in the middle of the kidney, for example. So it's really phenomenal what what the consortium has been able to do in pushing ahead these multi-omic single cell measures. Another big focus of this last round of ENCODE have been a lot of perturbational studies. So actually using technologies like CRISPR-Cas9 to go in and manipulate the genome and the epigenome to try to figure out what happens when you start to change things. That's the thing that's really come up. It's a technology that was developed sort of outside of ENCODE over several years and has really been embraced. And I say, that has been worked on very hard within ENCODE with the goal of being able to start to deploy those technologies much more broadly. And you're starting to see that now in kind of some of the intellectual successors to ENCODE. And I think on taking all of that data and bringing it together has required huge advances in how we actually interpret and analyze all those data sets. There's been a sort of an incredible push in developing new machine learning approaches, for example, to figure out what are the actual nucleotides that are contributing to all the signals that we see. And with this final version of ENCODE, you're going to start to see these very detailed single nucleotide precision maps of like, these are the key nucleotides that are driving this transcription factor binding site that's making this chromatin open and regulating this gene. So I think, as we're reaching the end, we're finally starting to put all these pieces together and starting to get a sense, not just of, where things are in the dark regions of the genome, but actually how they all interact and play on each other to start to drive gene regulation and ultimately to cellular phenotypes.
Gosh, that's so incredible. It's like so many different advances in genomics all being applied kind of towards the same problem, and the amount of expertise that you need, which you have as an investigator, to be able to not only analyze those data, but to understand them at a molecular level is really incredible and really an inspiration. As a follow-up question, as you look forward to what genetics and genomics looks like over the next 10 years, how does that understanding of gene regulation and transcriptional regulation, like how do you think that it will help us understand health and disease?
Yeah, I think it's a great question. I've been really fortunate. I've been sitting and really involved with the NHGRI, the Human Genome Research Institute's kind of future planning efforts, and now more recently I've been involved with their advisory council. So I've been getting kind of a sense of sort of where the field is going and in that regard and I think there's a couple like really salient themes that are certainly emerging. I mean, I think one is, while ENCODE has been and individual research projects and other consortium like ENCODE have really moved the needle in understanding kind of the landscape of the human genome. There's a huge amount of effort needed to understand all of the diversity of genomes that are out there, and all of the the different ways that that genetic influences from different ancestries and so on and so forth can all have different effects, for example. So I think that as we start moving towards disease, we're going to really want to make sure that we're moving away from a reference genome of a single individual, but starting to look at, for example, these pan-genomes that you're starting to see come out. And you're going to start to see a lot more, I think, genomic studies like ENCODE being applied to much more diverse populations and much more diverse samples. I think as we start to get that type of information and we have a much more kind of population scale understanding about how the genome functions, I think that's going to be the key point, sort of the turning point, where we can start to start to read out genome sequences, for example, right? So we can, ultimately a goal would be, right, to look at someone's genome, figure out what disease they may have or what disease they're most likely to have, right? And I think as we increase diversity within genetics, as we increase diversity within genomics, I think we're going to start to get the information we need to start to make that possible. I think that's one piece. There's many other pieces that need to fall into place, but that's certainly one of the major efforts that we see coming out of, for example, the Human Genome Research Institute and their funding and the projects from that. It's a major focus of some of the new consortia with that are akin to ENCODE moving forward. And I think it's an area where the field is going to be, everyone's going to be very well rewarded for the work.
That's great. Tim, I'm a clinician who takes care of patients who have genetic disorders, and sometimes we can't figure out what the genetic cause of their disease is. And I know that you're a principal investigator in the Duke Center for Combinatorial Gene Regulation, which is an NIH Center for Excellence in Genomic Sciences. Can you talk a little bit about the work that you've been doing with your SEGS grant and
Mhm.
what that kind of looks like for the face of how we solve these clinical problems?
Yeah, so that's been a super interesting project. It's been really amazing. It brings together 7 different PIs from Duke. It includes folks that are purely clinical in their research. It involves folks that are purely computational and statistical. It involves people that have only ever worked on cell lines in the lab, and it really brings all these people together in an effort to try to, as you say, connect the dots between some of these clinical diseases and the genomes and the underlying causes. So the SEGS itself is taken on a hypothesis that one of the reasons we're not being able to diagnose these patients is because we're only looking at one variant at a time. We might say, okay, it's this one variant, the other variant. Whereas we think there's a huge amount of missing information by not looking at how combinations of variants might act together. And those combinations can happen at different scales. It could be they have 2 genetic variants right next to each other that are somehow contributing to disease more than you might expect just from one. It might be that they have 2 variants that are very far apart, but the genes that they impact are themselves interacting with each other and causing greater effects, and so on and so forth. And so we've been really pushing hard both to develop new technologies to allow us to understand how these combinations of variants act together, and then very clearly keeping that close to the clinic. We don't want to just develop these technologies and have them kind of fly off into space. We want to make sure that we're bringing them back to clinical cases on a regular basis. And I mean, to some of the point you raised, we're working really hard on those clinical cases to focus on the patients that have really eluded genetic diagnosis so far, right? Patients that have been through the gamut of phenotyping and genetic testing and, in some cases even whole genome sequencing. And it's not really clear what's causing the disease that they may have, right? We've had some interesting successes early on. We've already found, I think, 3 or 4 patients where we've been able to pretty definitively define what is the actual cause of their disease, identifying new causes of these rare diseases. But I think one of the things we've learned along the way, right, is that every rare disease is special in its own way. And so one of the hard things that we've been really struggling with is, can we start to get some common themes? Can we start to put some principles in place that will allow us to make this much, much more routine? We've actually, much to our surprise, we went in with a hypothesis that there was going to be a lot of gene regulatory variation that we were finding. What we keep finding time and time again is splicing related variation, not something that we really intended to find, but it's also something that doesn't get really looked for nearly as much as you might expect with normal genetic testing. And so sort of spinning out of that SEGS project, there's been a sort of a whole new emphasis of our team on understanding how indeed changes in splicing are contributing to rare disease in ways that haven't been identified before. What's exciting about that is, when it comes to a gene regulatory element having a genetic variant, sometimes it's a little hard to think about how you might benefit that patient. But when we coming across these gene splicing related variants, there's already in place a whole suite of molecules that can impact splicing for genes in many, many different ways. And so there's often a really natural path to start thinking about, how you could even develop, clinical care based on this, right? It's always challenging when these cases are rare. You can't do clinical trials like you're normally thinking about it. But that's not stopping us from pushing ahead and at least trying to, develop the approaches and the analyses and the methods to get these molecules as far as we can, so that way, if indeed they can help these patients, we're ready.
That's so incredible, right? I mean, 2 really important themes. One is for a given patient and their family members to understand what the risk genetics might be, but also importantly, incredible work where you might actually be able to develop therapeutic advances. And this is a very important topic as we think about therapies in these rare and orphan diseases. So really, really incredible work.
I think what's been most surprising to me, though, is just how often we keep finding these splicing related effects. And so I'm I'm not sure what the future will bring. The next 5 cases might be completely different, but I am starting to sense that there might be a theme here that could at least give us a more, more robust pathway forward for these patients. They don't have to wait as long in the future.
Great. Well, thanks so much for your time today, Tim. Really wonderful work, and I'm really honored to call you a colleague in the broad Duke Genomics population. Thanks, Tim.
Great chatting with you.